Dr. Jander. And well, hey everyone, my name is Mohamed Elgallad. I was working in the BTI Computational Biology Center with Dr. Susie Strickler and Adrian Powell. And it was mostly like a conservation effort. And the title was Characterizing a Locus Linked to Beech Bark Disease. So I'm just going to give like a brief background about beech trees and beech bark disease and how it's like its population is declining pretty rapidly. And this is me, Dr. Susie, Dr. Adrian, and Isa, who helped us collect some samples from our knot-4s. So the project goals was to sequence New York susceptible American beech genome along an already sequenced resistant tree that we had sequenced. And we were going to compare, and we already compared the region containing markers associated to resistance to beech bark disease. We developed an expression atlas to search for significantly correlated genes of the gene of interest. And we developed some primers that will enable us to screen additional New York beech samples for resistance. So American beech or Fagus grandifolia, which means large leaves, and it's the only native species of beech in eastern North American deciduous plants, deciduous forests. And I guess everyone has seen like a beech tree before, like while hiking. It's mostly like, because it has like a really smooth bark that makes people like kind of carve into it, like, you know, like Jason, Trevor, Sally, and those kind of things, which I'm totally against. Please don't carve trees. Beech trees are slow growing. They grow up to like 300 years. They take their time, and they're shade tolerant, and they also could grow in low fertility soils. And they also have really high quality wood, which makes them really commercially like significant. And aside from all that, they have like a really important ecological importance because they provide a lot of species with beech nuts. And the species that eat these beech nuts range from birds to deers and black bears. And this is Dr. Susie labeling the tree that we sampled for DNA extraction, and this is a beech tree. So beech bark disease. Beech bark disease is a disease caused by an insect fungal complex attack that begins with a scale insect, Cryptococcus fagusuga, that attacks the tree to feed on the sap. And in turn, that like insect feeding creates like points of entry for the fungus, which is in the family of Neonectria, and there's 2 species known in North America. The most visible signs of beech bark disease, as you guys can see in the first 2 pictures, is this white waxy like secretion that scale insects kind of secrete, and it could be either like all over the tree depending on the population of the scale insect, or it could just be a couple dots depending on like the tree's tolerance and like the scale insect population. The second sign, which at this point, it means that the tree like really got like, it's pretty much infected with the disease. So this is called a bleeding spot, and it's caused by the fungus. And it's usually like circles that appear like it's supposed to be red, and that's beech bark disease. So there's a previous literature that really helped us in this project. It was by Kalik et al. And they did a GWAS with beech trees to screen for beech bark disease. And they used like beech trees populations from many states, but they left out New York State, which was like a promising opportunity for us in New York. They also didn't have like a complete genome. They only used like a European beech. So that was also like a downside for them. So we came in turn in our research. So the BC and Dr. Adrian and Dr. Susie already had sequenced the resistant tree, the resistant T8. And our project came in turn, where we were trying to sequence a susceptible tree to Beech Bark Disease. And we went to Arnott Forest, which is like 30 minutes from Boyce Thompson. And since beech trees are like pretty huge trees and they live pretty long, we had to get some leaf samples for DNA extraction. We had to kind of cowboy style, grab the trees with a hook. And this is Dr. Adrian cutting off some leaves, and this is Issa helping us retrieve some samples. Sometimes we were on the truck trying to grab the branches. This is the individual we actually sequenced, the S20, which stands for susceptible. And this is us in Arnott Forest looking for beech trees. And this is Dr. Susie. So the next step after retrieving the leaf samples was DNA extraction and library preparation. So we ended up doing 2 extractions. The first one, we did it with 2.06 grams, and we didn't get the concentration we needed with the qubit after testing it with the qubit. So we ran another round of DNA extraction, which was with 5.35 grams of leaves, and we got a pretty much good concentration. And we sent the samples to go through bioanalyzer to check for if the sequences have long reads, because we were going to sequence it through nanopore, which uses long reads. So in the step for the DNA sequencing, we used a nanopore min-ion. And what we retrieved, we left it throughout the weekend for about 2 days, and we found that the longest sequence was 87.5 kilobases. And the total sequence we retrieved was 21 gigabases. And the average kilobases we retrieved was 8.29 kilobases between and 17.5 kilobases, which was pretty good. The next steps was the genome assembly. We started with the sequencing, and after the sequencing, we retrieved a FASTA5 file that we used Guppy for base calling to change that file into FASTA-Q. And then we used Minimap and MiniASM for genome assembly. Then we used both Racon and Medaka for polishing that genome assembly. And we used a Busco report to check both the resistant and susceptible individuals we have for the quality of the genome assembly. And here are some of the reports we found. This is the sequence count we retrieved and the total length, the longest sequence, and the N50 length. And here's the Bosco report. So as you guys can see in the Bosco report, the complete Boscos we retrieved was more than 90%, which means our genome assembly was pretty successful into doing what we wanted. And the fragmented and missing, which are the bad parts, were really insignificant, which means we retrieved a really successful genome assembly. The second part of the project was analyzing the S20 sample. So we ran a couple blasts. We had obtained some markers that are linked to resistance from the Calic paper. And we blasted and compared those markers to the resistant individual that the BCBC had sequenced before. And there are 4 markers, and we compared them to the resistant genome, and we found the coordinates. And we did the same thing to the susceptible one. And the interesting thing is that we found 2 contigs, the UTG 30166 and the UTG 001671, and we found the coordinates. And as you guys can see from this circos, we mapped fragments of the UTG 000166 and the UTG 001671, which are the contigs in the susceptible one. And this is a fragment of chromosome 11 that has some of the markers, as you guys can see. What we found out is that there's a deletion in both contigs, and so if you guys can see this part compared to this part, there's a huge deletion, and you could also see it more in this contig. There's a huge deletion, which we also confirmed using structural variant PBSV. The next part of our project, we developed an expression atlas, and an expression atlas is basically like a heat cube. And we retrieved some RNA-seq from NCBI. These are the individuals on the top. We have the individuals in 3 seasons, so fall, spring, and summer of 2017. And these are the genes of interest. So we put the query gene, we run the expression, and it shows us the gene of interest and the frequency of expression. And it also shows us any genes of correlation to the gene of interest. And we could also retrieve some bar plots that shows us like the sequence, the frequency of expression, and it's color coded with the individual numbers. So why is this important to our project? It's important because this is our gene of interest, where the markers that are linked to resistant fall into, and we wanted to see if there was any correlation with that gene. Unfortunately, we didn't find any correlation between our gene of interest and any other genes in beech trees. But what we found was pretty interesting and promising, is that some individuals didn't really express that gene at all. Some expressed it in some specific seasons and not others, which would be really promising if we reach out to our collaborators and actually ask them to like screen for like Beech Bark Disease in these individuals. So the conclusion in future work, we managed to find a locus that is linked to resistance to Beech Bark Disease. And we found out that the susceptible individual contigs of the gene of interest contains a couple deletion mutations. And the structural variance data that we retrieved helped us to develop some primers that hopefully we're going to use in the future to screen additional New York beech samples for resistant. And our expression atlas data showed no strong correlation between our gene of interest and any other gene. For future work, hopefully Dr. Susie and Dr. Adrian are going to work on quick methods of DNA extraction from leaves in the field. And they will also develop, using the primers we made, loop-mediated isothermal amplification, or LAMP, which is basically a little PCR tube that they use in the field to like kind of screen for resistance or a specific gene. And this is me. So I'd like to thank Dr. Susie Strickler and Dr. Adrian for being wonderful mentors and helping me throughout like my research journey. And I'd love to thank BTI for this wonderful opportunity. Dr. Fei Wei, Dr. Eric Richards, Dr. George, thank you so much. Delaney and Olivia. Also Arnold Forrest, Peter and Britt for helping us over there. The Arnold Arboretum of Harvard University, BCBC, and Cornell University. Last but not least, Thomson Server for being great, helping me retrieve my data. And thank you so much. So resistance is pretty rare in like nature. I read like in the Calic paper that it's almost like 1%. So that's why it's like the disease is pretty dramatic. Like the population is like decreasing dramatically, especially with beech trees. Like they take their time kind of growing. They take like 100 years. So the disease is kind of like dramatically like decreasing the population. As for the resistant question, can you repeat that? Well, I think that's the plan for the future. So I think like after we like actually pinpoint the gene of interest, like we just know that this gene is actually responsible for resistance. They're going to use that data for like conservation efforts and also to help like woodlot managers to like start breeding like resistant lines of like beech trees. You're welcome. You could whoever. Yes. So to screen like little youngling like trees was the whole purpose of this project. So what Dr. Adrian and Dr. Susie are planning on in the future is after they like after they develop that lamp like technique or like tube, they're gonna use it for like future like conservation efforts through like they're gonna have like a lot of plants. So instead of just letting specific trees like grow, they're gonna test all of them for resistance, then grow the resistant ones. If that answers your question. Uh, we're not really entirely sure. So that's the next part of the research we're gonna work on. So the interesting thing is that some trees don't, like some individuals, they don't express it at all. Some trees express it, but we're not exactly sure what protein or what trait it expresses. So that's part of like future research we're gonna work on.
So we should probably move on in terms of time. We'll have a break shortly and then